Hi, everybody. My name is Kwan. I am a mentor and a co-director in the NMP program for sci-MI. Welcome to the Cognitive Neuroscience lecture. In this lecture, we will learn about how the neural structures govern our cognitive processes like attention, memory, languages, emotions, and so on. So let's define what cognitive neuroscience is, right? So cognitive neuroscience examines the biological processes behind cognition, like the mental action or process of acquiring knowledge, understanding through thoughts, experience, and the senses. And now you can think of cognition on the surface level as thinking, but thinking is a very limited view on cognition. A deeper sense of cognition is the brain's processing of information. So information processing. A metaphor that can be useful for you is to think of the brain, like the neural structure themselves, as a hardware of the computer, and cognition is the software that it runs. So it's the information processing. The only difference is that computers are universal machines and can run any software, but the brain software is more unique to the individual and more directly tied to the structure of the specific brain. Okay. To help you understand cognition a little better, let's compare it with two other levels of description. The neural processes, like the one from the molecular lecture, and the behavioral processes from the behavioral neuroscience lecture. In this case, we consider the scenario that someone is driving and then comes to a stop at a stop sign. So, at the cognition level or the mind, we're concerned with thoughts and mental processing. So, in this scenario, it might be when visual information from the stop sign gets processed and recognized as an object, and more specifically as a stop sign with a specific meaning. The meaning is retrieved from memory, interpreted, and then that's when you send a signal to your motor cortex and issue that stopping behavior. Okay? In the neural process, we are concerned with the brain and the body, so the physical and biological processes that implement cognition. So this is not the information processing of that visual information or the interpreting of it, but it will be concerned more with signal transmission from the retinas in your eyes to your visual areas in the occipital lobe in the brain, and then we may have selective firing of neural populations, synapses, etc. So in the same scenario, if we're looking at neural processes, we will be looking at different things. Finally, in at the behavior level or action level, we are looking at execution of a response to a stimulus. So, for example, stepping on the brake pedal when you see a stop sign, that is a behavior in response to seeing the stop sign. Now you can see that these three different levels, they are not the same thing, but they can describe the exact same scenario. Okay, and as like an introduction to the cognitive neuroscience lecture, I want to also touch on the philosophical stance that we're taking just by studying cognitive neuroscience, right? So there's a philosophical stance on mind and body problem, you know, which in which like cognitive neuroscience we study the relationship between the brain and cognitive processes, right? So there are two things here. One is the body, the brain, and cognitive processes we can think of as the mind. So this field would presuppose that the physical brain, right, the body, gives rise to cognition, the mind. So this is a philosophical view called materialism. It means that the material thing, the body, gives rise to the mind, and they are not like completely separate things. Furthermore, in cognitive neuroscience, we usually also take the stance of computationalism. So this stance believes that the relationship between the brain and the body, no, so between the brain and the mind is built on the brain's computation that is performed on mental representations. So we think of the brain like a computer that processes information, and that information is the mental representations that we have. Now, with that kind of intro and definitions in place, I would like to go through a little bit of the outline. We will go through the methods used in cognitive neuroscience, and then the rest of the lecture will be exploring different areas that cognitive neuroscience applies to. The main ones being attention, learning and memory, language, emotions, and because we don't have a lot of time in this lecture, uh, there are references that link you to additional resources, as well as um, in the final section, in the other areas, there are the names of other fields in cognitive neuroscience that, if you're interested, you can also look up as well. Now let's dive into the methods. There are four main groups of methods that I like to think about in cognitive neuroscience. All of these methods tell us something about how the brain affects cognition or how different types of cognition activate the brain differently. Okay? They vary in invasiveness, cost, portability, time resolution, spatial resolution, and permanence. You've seen some of these methods in the behavioral lecture as applied to animals, right? But in studying humans, the methods can be applied in very different ways than what you've seen before. So it's worth going through it again. We don't have time to go through everything here, but the list is there in case you're interested to look any of these up. Okay, so I'm just going to go through all these four quickly. Manipulation methods, we change the brain by activating or suppressing a part of the brain, like using magnetic field, electric current, or sometimes temporary drugs, and observe the change in behavior that results. This method goes from the brain to cognition. So we disable or enable the brain somehow and see how cognition changes, and it's usually temporary. Lesion methods, the second broad category, also go from brain to cognition. So, namely, we damage a part of the brain and see what cognitive function gets impaired. Now, in humans, obviously, it's unethical to do this for research purposes. However, if there's a naturally occurring damage like stroke, concussion, or like brain surgery that's essential to be done to treat conditions like epilepsy or cancer, researchers can take advantage of the need for lesions, detailed scanning, and other medical information that is gathered from that patient case with the patient's informed consent, of course, to study how these lesions affect the brain. So historically, this method originates in animal experiments, but it can also be done in humans in naturally occurring cases or necessary surgery. For the third broad category, neural recording, it works in the opposite direction. So we manipulate cognition by showing different stimuli or asking people to do different tasks. These tasks or perceptions create different activation patterns in the brain, which we can record and study. Controlled carefully, this analysis can be very informative and is usually the main way cognitive neuroscience is thought about these days. So these are things that you might be familiar with, like EEG, fMRI, etc. Okay, and finally, we have the modeling techniques where we abstract the cognitive processes into flowcharts, algorithm, or mathematical models, and then test whether that model make good predictions about actual cognition in humans or behavior. This method closely ties to computational neuroscience, which will be in next week's lecture, so we're not going to go too deeply into this method. Now, I want to go through and give you some examples within these broad categories, just so you have a better idea. Um, okay, so let's dive into some examples from manipulation. Again, manipulation methods when we change the brain temporarily and then observe change in behavior. So I think that you have already seen TMS in the behavioral lecture. So TMS achieves this brain manipulation with magnetic pulses that activates or interferes with the brain's natural electromagnetic activity. But something similar can also be done using electrical current, especially in humans. This is a technique that we call TDCS. This technique is often used in the medical field and is achieved by placing electrodes on the scalp Electric current is then delivered to the brain through these electrodes during some therapeutic activity. The idea is that by repeatedly stimulating areas that is damaged or is struggling to perform a function while the subject is also behaviorally performing that function as well, it would create long-term potentiation that leads to the brain area being activated even while the behavior is being done without the stimulation from the external source. It can be used to treat anxiety, depression, and some types of aphasia in humans. The next example is from a lesion study. So this is a method where we look at the overlap of brain lesions. Since in humans, researchers are limited to studying naturally occurring cases of brain damage, it can be hard to isolate a precise area that is responsible for a single function. Right? One way to get around this limitation, though, is to find patients with similar patterns of impairment in behavior and attempt to find overlapping lesions within that group of patients. The example study here finds the most overlapping area of damage in patients who are impaired in their moral judgment. So everybody is impaired in moral judgment through measured through any standard behavioral measure that they have. And then they can look at all the MRI scans of the patients, normalized and standardized to standardized brain, which is really popular. It's called the MNI brain. And then we can identify which regions has the most overlap. So that would be areas in red or orange in the graph on the right. So from this we can conclude that the area is likely necessary for the cognitive function of interest, namely moral judgment in this paper, but not necessarily sufficient, right? So it is damaged, and then you can't do moral judgment. It means it's required, but is this the single area that does moral judgment? That is not true. Okay. And just so you're better oriented in the image to the right here, each of the 7 images are not of different patients, but they are horizontal slices of the standardized brain MRI image at different depths. Okay. Now, in the lesion method, I also want to introduce the important term double dissociation, which is pretty crucial to a lot of research in this area. So, in double dissociation, we use this term in opposition to correlation and single dissociation. So in correlation, we can only say that 2 functions or 2 brain areas either tend to occur together or get impaired together, but we don't know if there are 2 steps in the same pathway, right? That's correlation. Now, if when we observe single dissociation, such as there's patient A and they can understand language but not produce it, then we can make a stronger claim that whatever damage patient A has, it affects language production. However, this is still not a convincing evidence that language understanding and language production are 2 functionally independent cognitive processes. Okay? Perhaps there are cases where maybe language comprehension is maybe just easier than production. So the area that is damaged make it so that the pathway cannot perform the more difficult task, for example, but they may be the same cognitive processes using the same pathway. However, if we found another patient that is impaired in the opposite direction, okay, for example, their language comprehension is impaired, but they can produce language just fine, then this is called double dissociation. Patient A and patient B together forms a pattern that tells us that language comprehension and production are functionally independent and they are computed using different brain areas. Okay? So with this, you can make stronger claims about the brain, stronger claims that brain area X is used for task A and brain area Y is used for task B, for example. And if you graph the performance, this is what you might find. So ideally, we want to have a control who performs very well on both of these tasks. And then we have two patients who performs well on one task, but poorly in another. And they should form an intersecting pattern like an X, as you can see here. Okay, let's move on to the third broad category, which is neural recording. There are so many neural recording methods, which is a good thing because some people have limitations where they are not able to undergo some of the neural recording methods. For example, if someone has an implanted cardiac pacer, they cannot be in a strong magnetic field, which is necessary in some of these methods like fMRI and MEG. Okay, so a common way to classify neural recording is by their temporal and spatial resolutions. An ideal measurement method would be at the top left corner of this graph here, which means it have high resolution in both time and space. But these methods tend to be invasive, which is why the other ones exist, even though their resolutions might not be as good. So spatial resolution, just to get through the terms here, is how precise the location of the signal we can measure. And temporal resolution is how much or how little delay we have between the signal and when we can record it. I'm going to go through a few of these neural recordings, for example, because this is a really prevalent method in studying cognitive neuroscience right now. Let's first go through the direct recording techniques. The best way to measure brain's electronic activity is to put electrodes directly on the brain surface, as in not outside the skull here, but open the skull and then place it directly on the brain tissue. Or to get even more accurate results, we can even penetrate the brain cells. Right? Okay. So in ECOG, the first one here, is a technique that puts electrodes array onto the brain surface itself under the skull, right? Is done only in active brain surgeries where the skull is already has to be open. So we're not opening people's skulls just for research purposes. The electrodes will sit on the surface of the brain so we can pick up good electric signal, especially for the surface or shallow areas. And it does not penetrate any cells, so it's not destructive. Other electrode arrays can penetrate into the extracellular matrix between neurons. So these goes a little bit deeper, but they don't penetrate the neurons themselves. And there are also some other types that can penetrate into the neurons themselves. These methods, though, are not used on humans usually because they are highly invasive. And once a neuron is penetrated, once you remove the electrode and you're done with the recording, the neuron will die. So that is so it's destructive, so it's not usually done on humans. But ECOG is done on humans in active brain surgeries. Okay. And then the next method is EEG. In EEG, we are measuring with electrodes on the surface of the skull. So outside of the skull, not under the skull, is non-invasive. Anybody can do it usually, but it can be a little like trying to decipher what goes on in a sports game by listening to the cheers from outside the stadium, right? So there's like that distance that the skull created here. And like with this method, we can tell when major cognitive events happen. In the sports metaphor, like if there's a goal, you can probably hear it even outside the stadium, but you probably can't tell exactly where it comes from. Um, and exactly what is being performed, etc. This method is also very noisy, as you can see in the image to the right, the rightmost image. It's noisy to the point where a single trial does not give us any information at all. The only way to read EEG is to average or aggregate across many trials many trials and potentially many participants as well. The method is also sensitive to movement, like muscle signals also use electricity and will also be sensed. And the other electrical activities in the room, like a flickering light bulb, could also introduce artifacts and noise into the data. Now, just to show you here, the image on the leftmost side is what EEG can look like. There are different types of electrode placements. So these are not like penetrating into the skull. They are just uh, contact nodes, and they'll usually put a little gel that conducts electricity right on the skull where the electrodes contact the skin for better conductance, but it is not invasive. The image in the middle is what you might see in research papers that does EEG. Now, the location here is very coarse, so EEG does not have a very good spatial resolution. So, if we see signals, high signals in the back of the brain in the occipital lobe, for example, it might not be as trustworthy as other recording methods. A common technique to use in EEG is called ERP, event-related potential, and this is when we average all these single trails, as you can see in the rightmost image. Each line presents the time course over the electrical activity over time of one trail. So with so many trails together, once we average them, the noise cancels out to be near the baseline voltage, near zero. And then we can see peaks and troughs more clearly. Okay, let's go on to MEG. MEG is a kind of like EEG, but instead of electrical activity, we're measuring the magnetic field, the change in magnetic activity. So electricity and magnetism inexplicably are like just very intertwined and they are linked together. So anything that we can measure electrical activity, we can also measure um, the inverse of it, which is the magnetic field. And this method have about the same temporal resolution as EEG, but a slightly higher spatial resolution. And the good thing, the advantage of this is that it works for deeper brain areas because the magnetic field is perpendicular to the skull surface. Okay. But as you can see on the image on the very left here, the machine is very big. It is much more expensive and less portable than EEG. Anything that is related to magnet and scanning and things like that usually tend to need like a cooling system, like an entire cooling system. So that is going to be very expensive and heavy to maintain. So it's not as portable as EEG. I also want to talk about, we just have 3 more recording techniques to go through. I also want to talk about the PET scan, the positron emission tomography. There are images of what you can expect to see here. Some of you might even have been in a PET scan before, either for the brain or for other body parts. So in a PET scan, we put some traceable molecules into the bloodstream, usually by like drinking juice that is the molecule is mixed into, and that will be absorbed into the bloodstream. And it exploits this fact that more blood flows to areas of high brain activity. So if, let's say, the visual cortex in the occipital lobe has high activity, then about 10 seconds later, the blood vessels is going to adjust to allow more blood to flow in to replenish the oxygen in the area with high activity that use a lot of energy. Now, we can detect the concentration of these molecules in certain areas to determine how much blood is being concentrated there. and that will correlate with the level of brain activity. So using this technique, we have good spatial resolution, but poor time resolution. And I quote here, there's also a small risk associated with the frequent introduction of radioactive compounds into the body. So these traceable molecules tend to be slightly radioactive, not to dangerous levels, of course. But if there's a longitudinal study that requires frequent scans, then that might be a risk. Okay. And then the next method is functional magnetic resonance imaging, or fMRI. fMRI is a bit different than just MRI, right? MRI is a type of scanning method similar to the PET scans, but it uses a quantum physics phenomenon called NMR, which also gives blood-related signal. So, except it doesn't need introduction of radioactive molecules in the body. It uses a very strong magnetic field to align the spin of electrons, use radio frequency waves to knock them out of alignment, and when the electrons realign their spins to the magnetic field, they emit radio frequency waves back that we can then detect and interpret into a 3D image. So it's not quite like capturing an image with a camera. Okay. MRI produces a static image. So just MRI, no F, right? MRI produces a static image of high resolution, usually like 1 by 1 by 1 millimeter per voxel. And voxel is like a 3D version of a pixel. So fMRI is a time series of MRI images. Okay. So to capture the images faster in a sequence, the fMRI usually have a slightly reduced spatial resolution. So instead of 1 by 1 by 1 millimeter per voxel, we have maybe a 3 by 3 by 3 millimeter per voxel. So without going into too much technical detail, fMRIs kind of give a higher signals when there's a lot of oxygenated blood in an area, which means it uses a lot of energy, which means there's a lot of brain activity. So it correlates with areas of high brain activity, just like the PET scans. And fMRI can be expensive, like over $1,000 per run, but it's highly desirable for research because it's non-invasive and there's, to date, no risk associated with undergoing the scans if you don't have like implanted metal in the body. Researchers are also required to do basic anatomical evaluations for brains of the research participants, which is a good way to detect any potential problems early. So, say a healthy person participates in fMRI research. When they get a scan, the researcher has to have a professional read the anatomical scan, just like in the medical setting, and see if there is any abnormalities. The brain can the machine, like the fMRI machine, can also be very loud and very sensitive to movement, so even a little fidgeting or head movement can mess up the scan a little. So the method has to be adapted if it is used with animals and children, which we've seen a bit in the behavioral lecture before. One key difference between fMRI for behavioral and behavioral neuroscience versus in cognitive neuroscience, which is in humans, is that even tasks without external behaviors, like just laying there in the machine, perceiving something, thinking something, or daydreaming, requires cognitive processing. So fMRI can also help us study these like behaviorless states. For example, like in resting state fMRI studies. Okay, there are also a reference here that you can take a look at is a technique in fMRI called diffusion weighted MRI, which helps measure the motion of water molecules and is used to map the brain connectivity. So if you're not interested in like which brain area has the highest activity when we're doing this, but you're more interested in which brain areas are connected to each other, then the DTI, diffusion weighted MRI, is going to be the topic for you. Now, final recording method, and I just want to throw this in here because it's a very new and exciting method being developed in the cognitive neuroscience field right now. This method is functional near-infrared spectroscopy, or FNIRS. Okay. It also measures blood-related signal, but this time using light instead of radioactive molecules or radio frequency waves, which are for PET scan and fMRI scans, right? So in this method, we also measure blood oxygen level, but not, but using infrared light instead. So it turns out that oxygenated and deoxygenated blood absorbs light, infrared light, differently. Something that could be like pretty obvious to us because the blood like appears different colors, right? They even make like arteries and veins different colors. It means that if we shine LED light, like the infrared light, through the skull and then measure how much light gets absorbed or comes back, then we get a signal for how much oxygenated blood is there, which again correlate with high neural activity. This method, and I just want to introduce it because it's such an exciting development because light is safe. LEDs and light sensors are cheap, so this is a very affordable method. And the signals are not as nearly sensitive to motion and noise as the other ones. As you can see, it can even be applied on children who are playing or doing actual activities, which is not usually true for methods like EEG, which can look similar, right? This kind of cap looking thing looks kind of similar to EEG, but in EEG, you have to sit still. There has to be as little motion as possible. Blinking or flickering lights or swallowing, things like that can cause some artifacts or noise in the data. But with FNIRS, the light is, you can block out the noise from the outside by just like using this black cap to prevent outside light from going in. And then it's not as sensitive to those noises. Okay. All right. Now, that was everything for the methods in cognitive neuroscience. Next, we're going to do a little whirlwind tour of the different areas that we can study in cognitive neuroscience. The first is attention. So, brain damage or lesion methods can be a window into studying attention. Simultanagnosia is the inability to perceive more than one object at a time. Patients are capable of identifying individual elements of a complex scene, but have great difficulty in understanding what is occurring overall within the scene. So, something like cannot see the forest or the trees, right? So, this is resulting from parietal lobe damage. Or another type of attentional deficit may usually occurs in stroke damage. So stroke damage to the right parietal lobe can result in unilateral spatial neglect. So that is the image to the right, bottom right corner of a clock. That is how a neglect patient might draw a clock. So they draw all of the numbers, right? All of the numbers, and they know that they kind of have to loop back around, but they kind of wholly ignore the left side of that image there. There are also dogs can also have this neglect symptoms actually. So old dogs that had a stroke may there are cases where they would eat only half of the food in the bowl. So the in this, if they have left lateralized neglect, then they might only eat the food on the right side of the bowl. And then it's not that they're full, so they don't eat the left side. If we give them another bowl of food, um, they will also like still eat just the right side, for example. Um, and then I would like you to, you have access to the slides. So I would like you to watch the video on the slides and follow the instructions in the video. And remember to pay attention. And this is going to be a video about a very um, common attention related psychological process. Um, but the since the video is not working here, I'm going to skip that and we are going to talk about memory. So, okay. Memory is often associated with the brain area we call the hippocampus, here, the classic center of memory. The hippocampus are depicted by the blue areas highlighted in these. The typical brain scan on the top row, top two images. Now, on the bottom two images, these are scans that show the damage in the brain of someone who has severe amnesia. So this is the amnesia patient's LSJ. And just so you know, in cognitive neuroscience, when working with specific patients, um, we usually use their initials uh, as an identifier to preserve their privacy. Okay, but in this case, the patient and their legal guardians and stuff like that has provided consent to share her image. So that's why I attached the image of LSJ here. Okay, so in the two bottom images, you can see that there are white areas, and these white areas are the damaged tissue. And if you compare it with the top row there, you can see that almost all of the hippocampus is basically decimated. And as a result, the patient has severe amnesia, both in forming new memories and in retrieving old memories. And here is a kind of a bigger picture of memory. You can see the memory is not really just located in the hippocampus, even though it is the site that is most associated with it. There are many types of memory. Sensory memory lasts on the scale of a fraction of a second. And then some information are lost, some information are retained in short-term memory, which is about less than 1 minute. And then some information, even a fewer amount, is retained for long-term memory. And within long-term memory, there are different types. There's explicit memory, conscious memory, things that I can declare, like what year it is, who is president right now, my biographical information, my age, name, address, things like that. Okay, and then there are implicit memory, which is usually used with procedural memories that's hard to describe, like riding a bike, skills like playing musical instrument, things like that. Now, um, So, just in conclusion, it's often believed now that the hippocampus is only involved in storing and retrieving explicit long term memory. So, it does not do everything, it is mostly concerned here. And in fact, patient Alice J, who we've seen in the previous slide, retains the implicit or procedural memory. So, she plays the viola, I think, and then there are experiments which show that she retains skills on the viola, making fewer mistakes on songs that she has practiced before. Okay. So, how to test for memory? So, there are multiple ways you can test for memory. I just want to give you a few examples. For explicit memories, they can use phrases or words. So they'll have a set of stimuli just like this. They present, this is called the Warrington Recognition Memory Test. So they will present these faces. There are 50 faces. They present one at a time for 3 seconds each. Sounds like a difficult test, right? So when you see a face, you have to respond whether that's a pleasant or unpleasant face, just to make sure that your brain is processing the visual image. And then right after the 50th face is shown, then there is a recognition test. So they'll show a pair of faces, like on the screen here at the top right. And then we must choose the face that was previously seen. Okay, so we can also do the same with words. It doesn't have to be faces. So of course, this is a pretty difficult task, and even normal adults are not perfect at this. There are 43 out of 50 is the average score. For LSJ, the patient with memory problems, we can see that both for faces and words, they have a score near chance. So chance level would be 50%, which is 25 out of 50. And that chance level comes from it being a two choice test, right? Now, the score of 28 out of 50 or 26 out of 50 is not significantly different from chance level of 25. So that means that they're most likely guessing and they did not remember any of the faces that they have seen before. This is one way to test memory. Okay, that's like for explicit memory. Now with working or short-term memory, we can use the digit substitution task. This is one of many types of tests that you can do. So in this task, you have to write the correct symbol under the numbers as quickly as possible in a limited time. You can only go one by one from left to right. You cannot skip. So good working memory allows you to keep some of these digit to symbol mappings in mind while you go one by one, which makes you go faster and get a higher score compared to looking back and forth from the box to the key legend at the top all the time. The resulting score is then compared to the average score for the same age group. So here, her score is 45. That's actually a little bit above average for her age group. So those are some ways that we can measure memory. Now, let's look at some molecular basis for it, right? So it is often believed that the Hebs rule, or this common saying, neurons that fire together wire together, is the basis for memory forming. So the more synapses occur, there is long-term potentiation, which means that there are more receptors and more of the neurotransmitter molecules, so that next time there is a synapse in the source cell, the target cells actually gets activated easier. So that's what it means by getting wired together. So that's memory. If you're interested in doing this field for the project, make sure you check out some of these references in the topic that you're interested in. Now let's look at language. Okay, so I'm just going to go through some language tasks that is possible to do for cognitive neuroscience. So often they will do things like self-paced reading. So self-paced reading is when you read a sentence with one word showing at a time. You try this in the assignment for this unit. Um, and then you yourself will press a button to bring the next word on. So this measures sentence processing speed and difficulty by recording the reaction time between button presses. So if I need a little extra time to process a word, I may take 50, 100, 200 milliseconds longer before I press the button to get the next word to appear. So this is a good indication of cognitive load for language processing. There are also fluency tasks like semantic or phonetic fluency, which means that within a time limit, you have to say either as many words related to a theme as possible, like you have 1 minute, say the name of as many animals as you can, something like that. That's semantic frequency, and semantic just means meaning. So we're saying things with similar meanings. Then phonetic fluency is phonetic is for sounds. So it's a similar task, but instead of a category, you're given a sound. So, like, say as many words beginning with the sound "f" as possible, right? Okay, so these are some tasks that can be done, and for each of these, you can localize which area is being used. Using an MRI, fMRI, for example, you can see like which area is associated with semantic fluency, phonetic fluency, and so on. Now, I would be remiss not to show you the classic areas that is associated with language. So these are the ones in the red box, right? So the Broca's area and the Wernicke's area. And the other ones, sorry, the other areas that are shown here is only shown as context. So these areas are found on the left side of the brain, the left hemisphere. And the Broca's area is usually believed to be the area responsible for the production of language and is close to the primary motor cortex. That's why they labeled it there. So patients who has damage in Broca's area will have something called Broca's aphasia. Aphasia is disorder in language. So they'll have um like someone who knows what they want to say, but they can't produce it. And then in Wernicke's area is often believed to be associated with comprehension of language. So it's close to the sensory cortex, and is usually responsible for the understanding. So patients with Wernicke's aphasia, or what we sometimes call fluent aphasia, can form sentences and produce language properly, but the words don't make any sense. The words don't make any sense, and they have trouble with comprehension. So if we deliver some kind of instruction like "please close your eyes," they might not be able to comply because they don't understand. These are examples of Wernicke's aphasia and Broca's aphasia. So let's look at Wernicke's aphasia first. Fluent but incomprehensible productions. Okay. You can watch the rest in your own time. It's quite fascinating. Now let's look at an example of Broca's aphasia.
Well, how are you doing today, Kent?
Serious. Okay.
Okay. Are you having pain anywhere?
No.
No? You feel pretty good in general?
Yes.
You can understand what I'm saying to you, right?
Yes.
Okay. Can you close your eyes for me? Perfect. Now tell me your name.
Because I've had no. I've had.
As you can see there, they have no trouble following instructions or understanding the questions, but sometimes have deep trouble finding the words or articulating what they want to say. You may also be interested in the field of language development. There's a lot there. Um, Babies start developing language from birth to around like 4 or 5 years old is when most of the grammar is in place. And young brains are very malleable. They can learn multiple languages, um, like better than an adult learning one new language usually, right? So there's a lot there as well that if you want to check out the review paper or the reference here, uh, it is a very interesting field in cognitive neuroscience. You could also look at emotions. Um, and emotions is a little bit trickier to measure because it is there's like some subjectivity to it. A common task is viewing faces with various expressions, right? Passively, either passively viewing it like in a scanner and see what types of brain activity can be measured, or they could have a task of either matching to another picture with the same emotion or naming the emotion, which then would include language. So, with all of these tasks, we can observe the brain response through recording techniques like fMRI. We can also measure emotions using various standardized scales like the Russell's affective space, self-reported responses, the Likert scale, or implicit measures like physical reactions like skin conductance, pupil dilation, and heart rate that is out of control. So those are going to be a little bit more objective, but they are limited in like this the subtlety of like the um, fine distinctions between emotions. So for emotions, the amygdala is often the classic center that is talked about for processing of emotion. Um, Fear, especially, drives the amygdala activation a lot. And there is this interesting case study of patient SM who has bilateral amygdala loss, which means both amygdalas are damaged or removed. And then this person had impaired cognitive ability. They cannot understand social distance. And interestingly, they were not able to make usual, like common everyday choices, like what to buy at the grocery store or what to wear uh, in a day. So we might think of decision making as like a rational process, but actually it is impossible to do without the emotional pathways in the brain. That might be another topic that you might want to check out for the project. And here are some other areas that you could explore. There's sensation and perception, social cognition, that is when there are more than one person. Um, Information processing of like social cognition is interesting because you have to not only process the information about the world around you, but also what the other person is processing or understanding. So there's like that meta level representation going on. Decision making, as I said, like we think of it as a rational process, but it use actually a system of different pathways in the brain to be able to do. We can look at consciousness and sleep, and meditation. I'll just throw that one in there. Resting state versus like activity, for example. There are studies of like how brains of expert works. So like expertise. So for example, like how does a person who is an expert at playing chess or solving the Rubik cube, how is their brain activity pattern compared to a beginner trying to solve it for the first time, for example? There's cognition in animals and babies. This one has large overlap with behavioral neuroscience, but it is extremely interesting. Because you have to be so creative in designing research methods to measure what you want to measure in animals and babies, since they can't report or understand instructions necessarily. Okay, and then I'll throw in a brand new field called cognition in plants and plants neurophysiology. Okay, so plants don't have neurons or neural pathways as we understand it, but they do have tissues and ways in which information or signals get transported and processed in quite complex manner. So you might want to check out this recent book. And if you would like to talk about it, I have read it, and maybe some other mentors inside me have read it also. So you can take a look at the research paper library and see who have read which paper, in case you're interested in any of them. And we have the reference here. You'll have access to the slides, so you can take a look at whichever one that you are most interested in. Thank you for going through the slides with me today. Don't forget to do the quiz, the short assignment, and I will see you in the other synchronous lectures.